Hello everyone and welcome to the new session of the biology lecture series, and today we'll be discussing the NCERT Exemplar problems from the chapter Sexual Reproduction in Flowering Plants. So we've already discussed some of the questions in the previous lectures, and today we'll be discussing the long answer type questions. So let's start with the first question. So here the question is starting with the zygote. Okay, so starting with the zygote, you have to draw the diagrams of different stages of embryo development in a dicot. Okay, so you have to just draw them. Okay, now let's discuss about this. You already know that zygote first give rise to the pro-embryo. Okay, and subsequently after the pro-embryo, subsequently which stages are achieved? Globular stage, then heart-shaped, and then you get the mature embryo that consists of the radicle and plumule and everything. Okay, so first you can say zygote, which give rise to proembryo. Okay, and then proembryo develops into globular stage first, and then heart-shaped, and then mature embryo. So let's have a look at the stages in the development of the dicot embryo. So these are the stages. Okay, so first here you can see in this image, number A, this is the zygote which is formed by the fertilization of the male and the female gamete. Okay, zygote is diploid. It has two chromosome sets. Okay, then what happens? Zygote divides. All right, there is a division in the zygote and it forms a large suspension cell and a small cell which is known as the terminal cell. Okay, here you can see the terminal cell. Okay, this is small and it is present at the antipodal region. Okay, so this terminal cell, okay, you can write about the terminal cell. So terminal cell, this is small and it is present at the antipodal end. So I'm writing A for the antipodal. Okay, and apart from this, zygote divides into one another cell which is a large suspension cell. Okay, here you can see suspensor later. Okay, so one terminal cell and one suspension cell, okay, which is present at the micropylar end. So this is the micropylar end. And later on what happens? You can see here, okay, there is more development here due to the divisions. Okay, one is the suspension cell which is large and the smaller one is the terminal cell. Then the, okay, here you can see suspensor cell. Okay, if we talk about the suspensor cell, it undergoes transverse division and it formed 6 to 7 celled suspensor. Okay, here you can see suspensor. This is formed by what? This is formed by the division in the suspensor cell. Okay, so if we talk about the suspensor is usually 6 to 10 celled. Okay, and it is formed by the transverse division of the suspensor cell. Okay, here you can see the suspensor, it consists of these cells. Then the uppermost cell or the first cell of the suspensor is known as haustoria. Okay, and the lowermost cell, okay, or the basal cell is known as hypophysis. Alright, so lowermost cell of the suspensor is known as hypophysis and the first or the uppermost cell is known as haustoria. Okay, here you can see after this suspensor, these both are the suspensor cell. This is the haustoria, this is the hypophysis. Okay, then later on what happens? Again you can see the embryonal cell, this divide twice. Okay, so embryonal cell divides twice, and okay, twice vertically. They divide twice vertically and once transversely, and they formed 8-celled embryo. Okay, they formed 8-celled embryo. Now, after this stage, after the E stage, you can see the F stage. Okay, then later on, okay, heart-shaped stage is developed. Okay, you can see this image G. This is a heart-shaped stage, and before heart-shaped stage, a globular stage is achieved. All right. Then what happens in the end? Here, this region is the suspensor. All right. Then what happens in the end? You can say this is the mature embryo, and mature embryo consists of the radical, primule, cotyledons, and here you can see the suspensor. Okay, so I've told you that embryonal cell, they divide twice vertically and once transversely, and they formed an 8-celled embryo. Okay, now 2 tiers are formed. Okay, first is the epibasal tier. Okay, first is the epibasal tier. This tier forms the cotyledons and the plumule. This tier forms the cotyledon and the plumule. Plumule will further give rise to what? Shoot axis. Okay. Then the other tier is known as hypobasal tier. Hypobasal tier. Okay. And this hypobasal tier will give rise to the radical. Okay, this will give rise to radical and what else? Hypocotyle. Okay, so here you can see plumule, you can see radical, you can see. Okay, radical will give rise to what? Root. Plumule will give rise to the shoots. Okay, here you can see the cotyledons. In dicot, 2 cotyledons are present. Here you can see a small suspensor. Okay, so these were the stages in the development of a dicot embryo. I will again summarize it. First, you can see here is the zygote. Zygote divides into suspensor cell and the terminal cell. Okay, so first, okay, we can say we have the zygote. This zygote divides and forms a suspensor cell which is large, and it is present at the micropylar end, and we have the terminal cell. Okay, this is present at the antipodal end. It's present at the antipodal end. Suspensor cell is present at the micropylar end. Okay, then what happens? This suspensor cell, it divides, okay, and it forms 6 to 10 celled suspensor. Okay, here you can see suspensor is formed. The first cell is known as haustoria and the lowermost cell is known as hypophysis. Here you can see in this stage is further development. Then a globular stage is achieved. Okay, after, okay, first a globular stage is achieved. Then after globular stage, you have the heart-shaped stage, and then finally you have the mature embryo. So you can say this is a mature embryo. Okay, this is clear. And this also I have told you, embryonal cell, they divide to form 2 tiers. First one is epibasal tier, second one is hypobasal tier. Epibasal tier will give rise to cotyledons and the plumule, and hypobasal tier will give rise to radical and the hypocotyle. Okay, so this is clear. Now let's move on to the next question. So let's start with question number 2. So question number 2 is, what are the possible types of pollinations in chasmogamous flower? Okay, chasmogamous flowers are those flowers that remain open, and the anthers as well as the pistil, or you can say stigma, they are exposed. Now you have to give the reasons also. Now let's discuss first about this. What is chasmogamy? So the pollination that occurs in open flowers, okay, means chasmogamous flower is known as chasmogamy. Or in other words, you can say pollination that occurs in chasmogamous flower is known as chasmogamy. It is the most common type of pollination in all types of flower. Okay, so this is most common, chasmogamy. Now, there are two types of pollinations. First is chasmogamy that takes place in chasmogamous flower. Okay, so first is self-pollination, another one is cross-pollination. So in chasmogamous flower, both type of pollination can occur. First is self-pollination, other one is cross-pollination. Why cross-pollination can take place? Because these flowers are open. Both the anther and the stigma are exposed to the external environment. So this can lead to cross-pollination. Okay, and self-pollination can also take place, where the pollens from the anther of the same flower are transferred to the stigma of the same flower. Now, let's talk about self-pollination. You already know self-pollination is also known as autogamy. Here what happens? There is the transfer of the pollen grain from anther to the stigma of the same flower. So this is known as self-pollination. It is found both in clastogamous as well as chemogamous flower. So clastogamous flower are those flowers that remain closed. So they remain closed. So that's why they will undergo self-pollination. The anthers and stigma are not exposed to each other. Okay. So it promotes self-pollination. Okay. Now, nitonogamy. What is nitonogamy? So it is a type of cross-pollination. We have already discussed about this. So it is the transfer of the pollen grains from anther to the stigma of another flower. And that flower is present on the same plant. Okay, so here pollination is happening. This is a type of cross pollination. Okay, and it is functionally a type of cross pollination involving a pollinating agent. So a pollinator or a pollinating agent is involved here. Okay, but it is very similar to autogamy. How it is similar to autogamy? Okay, because here what is happening? The pollens from the anther are getting transferred to the stigma of another flower, but that another flower is present on the same plant. Okay, means the genome is same or the genetic content is same. You can say genome, you can also write the genetic content. Okay, so these are same. That's why it is very similar to autogamy, self-pollination. Okay, now let's move on to next one. Next one is known as xenogamy. So xenogamy is completely cross-pollination. In this what happens? There is a transfer of the pollen grain from the flower of one plant to the stigma of another plant. Okay, so this is the difference between nitonogamy and xenogamy. Okay, here the pollens are transferred to the stigma of another plant. Okay, so this is only type of pollination which brings genetically different types of pollen grains to the stigma. Okay, now here you can see in this diagram is showing you cross pollination and the self pollination, how it happens. Okay, so here is plant A and here is plant B. So first let's study what is self pollination. Okay, so this is a figure showing you autogamy or the self pollination. Okay, here what's happening? You can see the pollens are transferred to the stigma of the same flower. Okay, so this is known as autogamy. And here in nitonogamy, what is happening? The anthers are transferred, okay, to the stigma of another flower. Okay, suppose from here to here or vice versa. Okay, and here a pollinating agent is involved. Okay, so this is a type of cross pollination. Okay, and then let's talk about xenogamy. So in xenogamy, what happens? Two plants are involved, plant A and plant B. Here is what is happening. The pollens from this flower, it getting transferred to the stigma of another flower present on another plant. And here is a pollinating agent. Suppose it's an insect or honeybee. All right. So this is known as xenogamy, where another plant is involved. So you can simply write here another plant is involved, or flowers of another plant is involved. Whereas autogamy and neonogamy, in both of these, what is happening? Same plant. Only one plant is involved. Okay, so this is clear. Now let's move on to the next question. Question number 3. Now with a neat and labeled diagram, describe the parts of a mature angiosperm embryo sac. Again, okay. So you have to describe and you have to label and you have to draw a diagram of the mature angiosperm embryo sac, and then you have to mention the role of the synergies. So this also we have already discussed how the mature angiosperm looks like, mature angiosperm embryo sac looks like. Okay. So before getting to the answer, let's study this. So megaspore is the mother cell for the development of the female gametophyte. Okay, and what is female gametophyte? Embryo sac. So embryo sac is known as the female gametophyte. All right, and female gametophyte is formed by what? It is formed by the megaspore. So initially what happens? You already know there is megaspore mother cell which is diploid, and it gives rise to what? Megaspore. Okay, which should be haploid. Now this megaspore give rise to what? Embryo sac, which is known as the female gametophyte. This is known as the female gametophyte. So if someone asks you what is a female gametophyte, so you can say embryo sac is the female gametophyte. And what is male gametophyte? Pollen grains. Okay, here what will happen? Meiosis will occur. Here what will happen? Mitosis. All right. So initially 4 megaspores are formed. Okay, 4 megaspores. Suppose you can write 1, 2, 3 and 4 megaspores are formed. Out of these 3 degenerate, and 1 megaspore it divide by mitosis and give rise to the embryo sac. Okay, now the nucleus of the functional megaspore divide. Okay, functional megaspore, this is the functional megaspore, this one. Okay, rest of the megaspore they degenerate. Okay, the functional megaspore divide mitotically to form 2 nuclei, which moves to the opposite poles, forming 2 nucleus embryo sac. Okay, now 2 more mitotic nuclear divisions occurs in 2 nuclei, okay, resulting in the formation of 8 nucleate embryo sac. So in the end we know that 8 nucleate embryo sac is formed, or you can say 8 nucleate and 7 celled. Why 7 celled? Because there is a fusion of polar nuclei. It is a fusion of 2 polar nuclei. Okay, that's why 8 nucleate and 7 celled embryo sac is developed. Okay, after the 8 nucleate stage, cell walls are laid down, leading to the organization of a typical female gametophyte, which is also known as the embryo sac. Okay, so this is clear. Now this is the image showing you the typical embryo sac of an angiosperm. So you can say this is a typical angiospermic. Angiosperms are flowering plant. Angiospermic embryo sac. Okay. Now antipodals. Antipodals are formed at the chalazal end. Okay. And egg apparatus is formed at the micropilar end. All right. So now you can see here, these are the two polar nuclei. Okay. Here is the central cell. Egg cell you can see. So egg apparatus is made up of the egg cell and two synergids. And within the synergid, filiform apparatus is present. Okay. So antipodals, okay, there are three cells and there are three nuclei. Okay. There are three cells and they have the three nuclei. Again, polar nuclei, there are two nuclei here. Okay, but when they fuse, they form a single cell. They form a single cell, also known as secondary nucleus. Then egg apparatus, here and all, if you talk about egg apparatus, it consists of two synergids. Okay, two synergids and one egg cell. Means again, there are three cells present in egg apparatus as well as three nuclei. All right. So if you talk about nuclei, three nuclei here, three nuclei here, 6 plus 2. In total, we have 8 nuclei. Okay. And if we talk about cells, 3 cells here, 1 cell here. Okay. 1 cell here. 4 cells plus 3, 7 cells. So it is 8 nuclei and 7 cell structures. Okay. So this is a typical angiospermic embryo sac. Now the mature embryo sac of the organism. Okay. Here, 6 out of the 8 nuclei are surrounded by cell walls and are organized into the cells. Okay. 3 cells are present towards the micropilar end, okay, which is known as the egg apparatus. Okay, and they are grouped together and they constitute the egg apparatus. The egg apparatus in turn consists of 2 synergids and 1 egg cell. This you already know. Now, there are 3 cells present at the chalazal end. Okay, and they are known as antipodals. Alright, now the large central cell is formed by the fusion of two polar nuclei. Okay, thus a typical angiospermic embryo sac at maturity consists of 8 nuclei and 7 cells. Now this embryo sac is formed from the single megaspore. So it is known as the monosporic embryo sac. Okay, this also we have discussed. From megaspore mother cell, okay, with the help of meiosis. What happens? 4 megaspores are produced initially, which are haploid. So these are diploid. After meiosis, which is known as the reductional division, these haploid tetrads of the megaspores are formed. Okay, then what will happen? Out of this, 3 will degenerate and the functional megaspore. So this is the functional one. This functional megaspore will undergo what? It will undergo mitosis. It will undergo mitosis to form the embryo sac. That will be what? 8 nucleated and 7 celled. So here you can see why it is monospermic, because it is arising from a single megaspore. Okay, single megaspore is giving rise to the embryo sac. That's why it is known as monosporic embryo sac. Alright, now let's discuss about the role of synergids. So second part of the question was you have to mention the role of the synergids. Okay. Now the synergids have special cellular thickenings, okay, at the micropylar tip, and these thickenings are known as filiform apparatus. Okay, so filiform apparatus they are present within the synergids. Okay, all right, and they are specialized cellular thickening. So you have to remember these things. And third thing you have to remember, it is present at the micropylar tip of the synergids. All right. Now, what is the role of filiform apparatus? Through filiform apparatus, the pollen tube enter into the embryo sac. Okay. So here, filiform apparatus which play an important role in guiding the pollen tubes into the synergid. Okay, and from the synergy, the pollen tube will enter in what? It will enter into the embryo sac. And then after entering embryo sac, it will release the male gametes, which can then fertilize the egg. Okay, two male gametes are released. So this is the role of synergids. Okay, so again, if we talk about synergid, first of all, you should say what is the role of synergid? It consists of filiform apparatus. And what is filiform apparatus? Okay, they are cellular thickenings at the micropylar tip of the synergid, which play important role in guiding the pollen tube into the synergid. And after that, when the pollen tube enters into synergid and it reaches the embryo sac, and then the synergid degenerate later on. Now let's talk about question number 4. Now here we have to draw the diagram of a microsporangium, okay, and label it walls layer. And we have to write briefly about the wall layers. Okay. So you already know there are 4 layers present. Now a typical angiospermic anther is bilobed, means it consists of 2 lobes. It is bilobed with each lobe having 2 theca. Okay. Now the anther is a 4-sided structure consisting of 4 microsporangia. It consists of 4 microsporangia that are located at the corners and 2 in each lobe. Okay, so this is the information you should know. Here you can see, okay, A. Image A is the transverse section of a young anther. Okay, so here you can see epidermis, okay, which is outermost layer. Then we have the endothecium. Inner to epidermis is the endothecium. Then here you have the middle layer. After endothecium, middle layer is present, and then the innermost layer is tapetum. So there are usually 4 layers here. Okay. First layer is the outermost layer is epidermis. Outermost layer is epidermis. Then you have the endothecium. Okay. And then we have the middle layer. And innermost layer is known as tapetum, which is the nutritive layer. It provides nutrition. Okay. Here you can see epidermis, outermost layer. Then we have the endothecium. Then middle layers are present. Okay, these are the middle layers. And then we have the tapetum, which is the innermost layer. And this is the microspore mother cell. Alright, again here you can see these are the pollen grains which are produced inside the bilobed anther. Okay. Now image B. Okay, this shows you the enlarged view of one microsporangium showing the wall layers. Okay. What is image C? Image C is the mature dehisced. Okay. There is a mature dehisced anther showing the pollen grains. Okay, so this is image C. Okay, here you can see the anther has dehisced and you can see the pollen grains. pollen grains have been released. All right. Now, in a transfer section, a typical microsporangium is circular in outline and is surrounded by 4 wall layers. Which 4 wall layers? We have already discussed: epidermis, endothelium, middle layer, and tapetum. All right. Now, let's talk about epidermis. So, epidermis is the outermost protective layer. It is composed of tangentially flattened cells. What kind of cells are present in epidermis? Tangentially flattened cells. Now the cells are closely fitted and have thick walls, which is helpful in the dehiscence of the anther. Alright, this was about the epidermis. Then second, inner to epidermis is the endothelium. So it is present below the epidermis and expands radically with fibrous thickenings. At maturity, these cells lose water, okay, and they contract and help in the dehiscence of the pollen sac. So this is the role of the endothelium, which is present below the epidermis. Alright, then we have the middle layers, or you can say middle wall layers, or simply you can write the middle layers. Okay. Now it is present between well-marked endothelium and the tapetum. Okay, so middle layers or the wall layers are present between the endothelium and the tapetum. These are thin-walled. Okay, these are thin-walled layers arranged in 1 to 5 layers, which also help in the dehiscence of the anther. So what is the function of wall layer? They help in the dehiscence of the anther and release of the pollen grains. Now the innermost layer is known as the tapetum. Okay, so it is the innermost wall layer with large cells. They have thin cell walls, they have abundant cytoplasm and have more than one nuclei. Okay, so this is a characteristic feature. They have more than one nuclei. And what is the function of tapetum? Tapetum is a nutritive tissue which nourishes the developing pollen grains. So you have to, okay, you should know the functions of these layers and you should know the structure or the type of cells which are made, whether they are thin walled, okay, and whether abundant cytoplasm is present or not, and how many nucleus are present. This you should know. All right, now the center of the microsporangium. It consists of the sporogenous tissue. So in the figure, you must have seen sporogenous tissue. Okay, so these sporogenous tissue undergo meiotic, yani ki meiosis, which is known as reductional division, to form microspore tetrads. Okay, so sporogenous tissue should be diploid. So then only meiosis can take place. Okay, so meiosis takes place in sporogenous cells and they form microspore tetrad. And this process is known as microsporogenesis. Okay, formation of microspores, okay, with the help of meiosis is known as microsporogenesis. This is clear, and microspore will further form pollen grains. Okay, and pollen grains will further, okay, form male gametes, okay, which are released inside the ovule. And then they will enter the embryo sac and they will carry on the fertilization. Alright, so this is also clear. Now let's talk about question number 5. The embryo sac of some apometic species appear normal but contain diploid cells. Okay, means 2n. Now suggest a suitable explanation for this condition. Alright, now let's have a look at the answer. A replacement of the normal sexual reproduction by asexual reproduction without fertilization is called apomixis. Or simply you can say, what is apomixis? We have already discussed. It is the development. Okay, it is the development of what? Seeds. Development of seeds or production of seed without fertilization. This is known as apomixis. And apomixis is a kind of asexual reproduction or asexual method. Okay. because here no gametes are produced, there is no meiosis happening here. Okay, so replacement of the normal sexual reproduction. So through normal sexual reproduction also seeds are produced. But during apomixis, what is happening? During apomixis, there is development of seeds, but this development of seed is without the fertilization. Okay. Example, replacement of the flower by bulbils and replacement of seed by a plant. All right. Now, apomictically produced offerings, okay, as it is a kind of asexual reproduction. So what is the feature of asexual reproduction? One parent is involved. Okay, if we talk about the feature of asexual reproduction, uniparental condition is observed. Okay, and what happens? The offerings produced are genetically similar to each other. Okay, they are genetically identical to each other. Okay, as well as with their parent. Okay, as well as with their parents. Okay, so apomictically produced offerings are genetically identical to the parent plant because there is no recombination and crossing over here. There is no chromosomal segregation also. Okay, why they are genetically identical? Because there is no recombination. There is no recombination. There is no chromosomal segregation. All right. So there is no variation. Okay. So the offerings are genetically identical to the parent plant. Now, in flowering plants, apomixis is used in a restricted sense to mean angiosperm, that is asexual reproduction through seeds. Okay, apomixis is used in a restricted sense. Okay, now in some plant species it is common. Okay, for example, in members of Asteraceae and Poaceae. Okay, so Asteraceae, like sunflower belongs to the family Asteraceae. Okay, and grass family, which is known as the Poaceae. So most of the members are monocots here. So members of Poaceae are usually monocots. In some species, the diploid egg is formed without reductional division. Okay, and develops into embryo without fertilization. This also we have already studied. Now it is an asexual reproduction in the absence of pollinators, such as in extreme environmental condition. So this is also a kind of asexual reproduction, formation of diploid egg cell, okay, without the reductional division, and then this diploid egg cell will give rise to the diploid embryo, and then this diploid egg cell will give rise to the diploid embryo. So this is the kind of asexual reproduction. Now in some species like citrus, okay, some of the nucellar cells surrounding the embryo sac start dividing. This also we have studied. Okay, so nucellar cells are usually diploid. They are diploid. So they undergo or they start dividing and they develop into the embryo. So this is also kind of asexual reproduction. Okay, so this occurs in the megaspore mother cell. Okay, it occurs in the megaspore mother cell which does not undergo meiosis, thus produces diploid embryo sac through mitotic division. Okay, here mitosis is occurring and there is no meiosis. So the cells of the nucellus which are diploid, they directly give rise to the embryo sac. So all the cells in this embryo sac will be diploid, which will give rise to a diploid embryo. Alright, so this is clear. So thus it explains that embryo sac of some apomictic species appear normal but produce diploid cells. Okay, they are producing diploid cells. There is no meiosis happening here. Okay, here you can see the nucellar cell is giving rise to the embryo sac which is diploid, and the embryo sac then forms the diploid embryo. Okay, this is happening with the help of mitosis. So this was the end of this lecture. Hope you have gained a lot of knowledge from this, and we will discuss the questions for the rest of the chapters in the next section. Thank you very much for listening.